Okay, welcome to our next speaker. I'm thrilled to introduce Yi Tang from UCLA. And Yi is the Chancellor Professor at Department of Chemical and Biomolecular Engineering, and also holds joint appointments in the Department of Chemistry and Biochemistry and Department of Bioengineering. So Yi has undergraduate and PhD degrees in chemical engineering from Penn State and Caltech. And he, after getting his PhD, he did postdoc training in chemical biology with Keaton Kostler at Stanford, and then joined UCLA in 2004. And Yi is, a true expert on not in genome mining, but really the biochemistry of fungal natural products. And we're thrilled that he's able to join us today. The title of his talk is Genome Mining of Fungi for New Natural Products. So please, Yi.
Okay, thank you very much, Nigel. Hi, everybody. It's a great pleasure for me to be here to tell you a little bit about what my lab does. For those of you who attended the previous talk by Professor Turgeon, you would gotten an introduction on fungi and the importance of natural products. So in this talk, hopefully I'll tell you the good things that fungi can do for both crops and other applications. I also want to acknowledge my lab first in case the time runs out. Shown here in bold are the students and postdocs that have been involved in the projects I talked about. And the funding for my research has been supported by NSF, NIH, and recently by USDA in a couple projects that you'll hear about today. So our lab for the last decade or so has shifted to using genome-based approaches to discover new natural products. And this is, in my opinion, has brought a renaissance of natural product discovery and really take natural products towards the interface of chemistry, biology, as well as synthetic biology. So the schematic here basically shows that from genomes, we can obtain blueprints of the microbes or plants, and then you can use bioinformatic approaches to find biosynthetic gene clusters. And then one can use different approaches to elucidate the natural product that's encoded in the biosynthetic gene cluster. And our focus has been fungi because of their resemblance. They are eukaryotic microbes, so a lot of their natural products do hit eukaryotic targets, and that's important for discover both herbicides, insecticides, and also human therapeutics. So our motivation is that upon the sequencing of the first genomes, some by JGI, some by the Broad, it was found that many of these classic Hall of Fame, if you will, hosts that produce very well-known natural products such as penicillin and lovastatin. They encode much more natural products based on their biosynthetic gene cluster than what's produced in the culture. So you can consider that all the fantastic drugs and useful compounds that have been discovered from fungi, it only represents a very small amount of what these fungi can truly produce. And if we can tap into the other cryptic pathways, we should be able to discover much more new natural products for different applications. Okay, so our lab uses different approaches to discover natural products using genome approach. For example, we use, if you consider in fungus, the biosynthetic pathway, BPGs are biosynthetic pathway genes. Here shown are 3 genes. They're controlled by separate promoters. These promoters are controlled by a transcription factor that's separately controlled by a different promoter that's subject to control by global conditions or epigenetic modifications. So if we can turn on this transcription factor to directly upregulate the transcription of these genes, then you can turn on a otherwise silent pathway. Nancy Keller and Christian Harwick and many other labs have done this. Shown here is one example. We did this to obtain new natural products from a marine fungus. We've also performed epigenetic modifications to turn on the transcription of otherwise silent biosynthetic gene clusters. In this case, we inactivated a histone deacetylase protein, and that led to dramatic changes in chemical phenotype of the host. And then we've also, main tool in our lab is heterologous expression, is to take these pathways completely outside of their natural context, put it into either model fungus or yeast to produce these compounds. So the one question then, as more and more genomes are being sequenced at JGI, Broad, and other private labs or other countries, the number of clusters are exploding exponentially. And shown here is a vastly outdated table of biosynthetic gene clusters in fungi, taken from 2016 from my colleague Maureen Hillmeyer's paper. I so far have not seen an updated table, and if anybody is aware of such a table, I would love to update this slide of all the different type of pathways in fungi. And what's important is that of total 9,000 around 2016, 2017, only 3% have been associated with known compounds. And this number is truly expected to be much bigger now, but I believe that the 3% characterized pathway should remain pretty much the same. So which means that we have many more uncharacterized pathway to be explored. So then the question really is, how do we search through genomes for gene clusters of interest, and how do we find natural products with new structures and biological activities? So today I want to tell you about how my lab thinks about these things. We come from both the chemistry side and also from the biology side. From the chemistry side, we want to prioritize the biosynthetic gene clusters based on potential structural novelty. And from the biology side, of course, we want to prioritize by potential new biological activity. So let's talk about the chemistry side first. So what makes the cluster novel? I think that's the question we all want to be able to reliably answer every time a new grad student comes in looking for a project. The way we think about it is that an unusual cluster should lead to novel natural products. And what makes the cluster unusual then is that first we should have very little idea what the cluster can make. As more biosynthetic gene clusters have been associated with products, it becomes now possible to predict possible products, but we want to look for those that cannot offer only minimal clue to the type of natural product. In fungi, the clusters are generally pretty small. So if we can find a large biosynthetic gene cluster, that can often lead to interesting products. Abundance of tailoring enzymes such as P450s, redox enzymes, etc., indicates extensive structural morphing. Hypothetical proteins, domains of unknown function. Atypical core enzyme arrangements. Core enzymes include polypeptide synthase, non-ribosomal peptide synthase, terpene synthase, etc. And then also combinations of different core enzymes in a single cluster, indicating more structural diversity. And something that we are working on very actively now is look for clusters that have no core enzymes. And that's not quite ready to be presented here, but hopefully I will be able to share with you guys in the next meeting or the meeting after that. So here, I want to just focus on combination of two or more coenzymes lead to complex structures. So shown here are some examples my lab and others have worked on that derive from biosynthetic gene clusters that contain more than one core enzyme. So for example, lovastatin contains 2 polypeptide synthases. This chateauviridine contains 2 polypeptide synthase. Cyclosporine, a very famous immunosuppressant, contains a non-ribosomal peptide synthase and a polypeptide synthase, etc. So what we think is that if you have more than one core enzyme in a fungal biosynthetic gene cluster, it's possible to discover new products. I want to start by showing some examples. The first example I want to show is derived from this fungus called Trichoderma Harzianum T-22. This is actually a beneficial fungus, so it helps the plant, it promotes plant growth in soil. It resides in the rhizosphere or near the roots. So if you look at the potential interactions in the soil, you can imagine a beneficial fungus can limit the growth of pathogenic fungi. It can help the root acquire nutrients such as iron. It might even kill certain weeds. And many of them we hypothesize can be facilitated by secondary metabolites. So this fungus is a commercial biocontrol fungus, so it's applied to soil. You can buy it. And when this fungus was sequenced, we find that, as expected, it contains around 65 biosynthetic gene clusters. And just as I mentioned before, less than 10 natural products have been discovered from this fungus. So it's really amazing to think that we are applying this fungus to soil, yet we know very little about the metabolites that can potentially be produced. But on the other hand, if we can access the metabolites encoded in all these gene clusters, we might be able to produce these metabolites in a more controlled way and directly apply the metabolites that can be beneficial to the plant instead of applying a fungus. So we started a campaign to do genome mining of pathways in this organism and try to identify compounds that might be beneficial to agriculture. We focused on pathways that contain more than one coenzyme first. So in this example by a previous postdoc, Wenbing Chen, she focused on a pathway that contained two different polyketide synthase, one hydroreducing, one non-reducing. What's also interesting in this pathway is this pseudo ACP enzyme, acyl protein. So for those of you who are connoisseurs of polyketide synthase, this pseudo designation basically means the active site in which the acyl protein is phosphopentathionylated is mutated. So it's actually not a functional acyl protein, hence named pseudo. But it's fused to a methyltransferase protein that is active, suggesting that this pseudo ACP might play some structural role in the function of this methyltransferase domain. So we performed a transcription factor overexpression in the native host. In the control host, the wild type strain, in their PDA growth, the lab mainly produced this aromatic polyketide called pachybasin. But when we overexpressed this transcription factor, we observed the production of two new compounds, this orthohydroxyquinone compound 1 and its oxidized version, this orthoquinone compound 2. And we did some electrochemistry on this compound to show that the redox potential of this compound can be sufficient to reduce iron 3 down to iron 2, which suggests that this compound could, it might not at all, but it could have a role in reducing the unavailable iron 3 in the soil to the more available iron 2 for plant uptake or for fungus uptake. So we did an experiment in which we took a string of Arabidopsis that's mutated in, that's decreased in its ability to acquire iron. And we grew the Arabidopsis either in iron deficient media. And when we grow it in the presence of EDTA, which is a positive control to help chelate iron, you can see the plant growth quite happily with a green color indicating a lot of chlorophylls. But if we grow it in presence of methanol without any methanol control, without any chelation agents, you can see the plants suffer chlorosis, which is a lack of iron. However, if we added equal amounts of this compound we just discovered, Tricholignan A, the hydroxyquinone version, you can see the color is restored. And upon looking at chlorophyll concentration, you can clearly see that this compound indeed is helping the plant acquire iron under iron deficient conditions and restore chlorophyll concentration. So this could be potentially one role of this compound. By the way, this is a new compound, haven't been isolated in nature before. So this could be a potential role for this new compound in helping plant grow. The biosynthesis, which I'm not going to elaborate, is shown here. Basically, the two PKS, one makes the diene, the second takes the diene and generates the 2,4-dihydroxy benzoate, and then further modification, including methylation by the pseudo-ACP-MT that gives this particular methyl group carbon 5. Okay, so the role of the pseudo-ACP, just one quick slide, what we think is that it's helping to recruit the methyltransferase to the polyketide synthase assembly to allow it to methylate this alpha position during polyketide chain extension. So we think the pseudo-ACP, as I mentioned earlier, because it cannot be phosphopentathiolated, serves as a provides protein-protein interaction to bring the methyltransferase to the ketosynthase. So having discovered the pseudo-ACP-MT construct, we decided to look for other biosynthetic gene clusters that might contain such a feature, which might lead to new compounds. So when we did perform genome mining of biosynthetic gene clusters in fungi that have pseudo-ACPs, we found another core dual polyketide synthase cluster that contains a pseudo-ACP, this time fused to a thiol esterase domain shown here. Now, these are not found in soil bacteria, fungi, but more found in insect, endosymbiotic insect, that are in symbiosis with insects, such as this mycorrhizal and mesoply fungus. It contains HRPKS, NRPS, and a pseudo-ACP-TE. Again, the pseudo-ACP is indicated by the mutation of the active site to something different from the canonical DSL motif. This cluster is found in many different fungus, so we decided to use Heterologous expression to express these three genes to see what kind of compounds are produced. And when we transfer this pathway into Aspergillus nigrans, which is our model host, we were shocked at the beginning to see tremendous production of one particular compound 1, along with compound 2, 3, and 4. When we solved the structures of these compounds, 1 turned out to be this furocarboxylic acid, and 3 turned out to be olivatoic acid. Compound 1 is the 8-carbon version of olivatoic acid, which contains a 6 carbon. I was initially not particularly excited about these compounds because these are just 2,4-dihydroxybenzoic acids, but as one of my students pointed out, that this is how we make wheat components. So it turns out that olivatoic acid is the precursor to cannabinoids. So in plants, 2 enzymes, olivatoic acid synthase and olivatoic cyclase, makes olivatoic acid, which can then be granulated and then cyclized into cannabinoids. And to this date, this has been the only pathway that has been found to produce olivatoic acid. And all commercial efforts to make olivatoic acid rely on the use of these 2 enzymes. Hence, there are significant IP restrictions. However, using our genome mining methods, we were able to find other fungal pathways that can also produce olivatoic acid. For example, one of the pathways we found produce exclusively olivatoic acid. This particular pathway in M. anisopoli makes tremendous amount of this longer version of olivatoic acid. And it turns out that the cannabinoids derived from this longer version of this 2,4-dihydroxybenzoic acid is actually one of the most potent cannabinoids known in inhibiting the CBD1, CBD2 receptors. So we feel with this platform, we have a different way of accessing these important precursors to make not only known cannabinoids, but also rare cannabinoids that might become important therapeutics in the future. Another quick example is genome mining in A. flavus. This is also a very important fungus that's used. That's a pathogenic fungus that colonizes the different plants. It has a lot of biosynthetic pathways. Again, only very few of them are known to produce compounds. And we focused on one particular gene cluster that contains 2 core enzymes, 1 non-ribosomal peptide synthase and 2 terpene synthases, a type of arrangement that's only being associated with one other product, alkylamine A, in the literature. So we're curious what kind of compounds can be derived from this type of pathways. And when we move this pathway into nitulins, we were very pleased to find it makes a very novel chemical scaffold in this compound we named flavonoidine. It contains a oxidized terpene scaffold fused to a 2,5,5-dimethyl pipercolate, as well as a carbon-nitrogen bond fused to this dimethyl cadaverin. For those of you who know terpene natural products, you know that carbon-nitrogen bonds in terpene are extremely rare, and this represents one of the very few examples. This is a related compound that was found previously. You can see that it's similar to our compound, but it lacks this dimethyl pipercolate. But so this is a testament to the type of chemical structures that one can obtain if you start with a novel biosynthetic gene cluster. But at the same time, it frustrated us because we tried many attempts to map out the biological activity of this compound to no success. It looks complicated. It must have some biological activity, but so far we have not been able to locate the biological activity. But in fact, this is actually not surprising based on our approach, is that we are looking for chemical diversity from the biosynthetic gene cluster, not necessarily biological activity novelty. So this leads me to my second point, is how do you prioritize by new biological activity? And this is something that I've talked about before, and I'll recap here briefly along with one new example, is that we've been using the presence of self-resistance enzymes, SREs, to guide us in looking for new biosynthetic gene clusters. So the question, again, coming back to this table, is that in the sea of new biosynthetic gene clusters, how can we search for gene clusters based on a desired biological activity? And if we can do that, that effectively bridges classical natural product discovery, which is bioactivity guided, and this new genome guided natural product discovery to discover more useful compounds. So how to mine natural products guided by biological activity? So if we consider what producing organisms of natural products, the purpose of the natural product, a majority of these natural products are what we can consider as toxins. They go into the invading organism and inhibit the function of some housekeeping enzyme in a invading organism. And when that happens, that limits the growth of the cells or causes cell death. But at the same time, if both organisms are fungi, then the producing organism also has the same housekeeping enzyme. And if that's inhibited and unmitigated, that'll lead to suicide of the cell producing organism. Nature is far smarter than that. So what nature has done is that it can also encode a self-resistance enzyme. These enzymes come in many different fashions. They can be transporters, they can be antidotes, or they can be an enzyme what we think are perform the same function as a housekeeping enzyme, in this case converting A to B, but is not inhibited by this natural product, hence have resistance to the natural product. But in the invading organism, no such resistant enzyme exists, so the cell will remain limited or die. And this self-resistance enzyme provides the essential resistance to the natural product before it is exported. So it's essential for the survival of the producing organism. It's frequently mutated, but it's not dramatically different, which means that bioinformatically, it will still have the same predicted function as the housekeeping enzyme. And it's, in our experience, between 50 to 70% sequence homology. And perhaps most importantly is that it's often co-localized with the biosynthetic gene cluster. So it can actually provide a very useful window, predictive window to what the cluster, the compound derived from the cluster, how the compound would function based on the predictive function of a putative self-resistant enzyme. These examples are abundant in the literature. So for example, fumagillin, a compound of which we work out the biosynthesis, and Jun Liu in the late 90s worked out its target to be human methionine aminopeptidase. And the gene cluster from Aspergillus fumigatus that produces fumigillin contains 2 resistant copies of methionine aminopeptidase. Ralph Mueller in worked on this compound called gracilimycin, a very classic natural product, and the target of which has been unknown for a long time. But upon finding its biosynthetic gene cluster, Ralph predicted it will target DNA polymerase sliding clamp because of presence of resistance gene, and that was validated using biochemical and structural approaches. Then our idea is that can we reverse this thinking and start with a target? We look for a biosynthetic gene cluster that has this resistance gene along with other core enzymes. So then that can lead us to infer the biosynthetic gene cluster will produce a compound that can hit the target that we are desiring. And then we can use different ways mentioned before to produce natural product, validate the target, and then this can be followed by a more traditional medicinal chemistry or a structural diversification to improve the activity.
You just 2 minutes, please.
Okay, sure. Thank you. And so we focus our initial effort on discovering a herbicide, which I will skip this slide. Our target initially, this is the branching amino acid biosynthetic pathway, and this is well conserved in fungi and plants. So we hypothesized that if fungi were to produce a compound that inhibits one of these enzymes, we can use it as a herbicide lead. At the same time, the fungus must also be able to resist this compound because this pathway is also essential for its survival. We focused on the third enzyme, dihydroxyacid dehydratase, which has no known natural product inhibitor, no effective inhibitor, and no crystal structure of this enzyme is known either, which is surprising. This enzyme is called dihydroxyacid dehydratase, short is DHAD. So we found one biosynthetic gene cluster that contains 3 enzymes, terpene, cyclase, ASTA, ASTB, CRP450s, along with a second copy of dihydroxyacid dehydratase. It's about 60% identity to the housekeeping enzyme. So we hypothesized that these 3 enzymes here will make a natural product to inhibit the housekeeping DHAD, while ASTD will provide the self-resistance. To make a long story short, we put this pathway into yeast. We produced a compound called aspartic acid. This was a known compound, but the biological activity of this compound was completely unknown. But we then hypothesized that this compound should inhibit dihydroxyacid dehydratase. Indeed, when we assayed in vitro using purified dihydroxyacid dehydratase, this compound potently inhibited both the housekeeping version in the fungus as well as the Arabidopsis dihydroxyacid dehydratase with Ki of around 300 nanomolar. But towards the resistance enzyme ASTD, it has absolutely no activity beyond within the solubility limit. So the resistance enzyme is indeed insensitive. We solved the crystal structure of this compound in the active site of dihydroxyacid dehydratase, shows the biochemical basis for the inhibition. But more importantly, when we spray this compound onto Arabidopsis, we can see very effective inhibition of plant growth, and it's effective towards a variety of different plants. And we're now in the process working with a startup company to evaluate, to do field trials with this compound. And lastly, in the last 30 seconds, I just want to share with you a recent discovery in which we also use this method to find a natural product inhibitor of the first enzyme in this pathway, this acetylactate synthase. This is the most heavily targeted enzyme for herbicide development with all kinds of synthetic compounds, but no known natural product inhibitor. And we thought this seems unlikely given its importance. Evolution should have produced a natural product to inhibit this particular enzyme. We go back to THT22. We think that we look at the genome, and we found one particular cluster in THT22 that seemed interesting. One compound THT22 makes is this compound called Harzianic Acid. It has been demonstrated to have antifungal activities, among others, but the basis of its activity is not known. The biosynthetic pathway is shown here, which led us to find a biosynthetic gene cluster consistent with this pathway. I'm not going to go into the details, of this pathway here. What we found in the pathway is that there encodes a second copy of this acetylactate synthase in addition to its housekeeping copy. In the DHAD22 pathway, this second copy is actually not clustered, but it's somewhere else on the genome, but it also contains 2 copies. But in these other homologous clusters, it's actually in the genome, which tells us that perhaps this compound can inhibit this acetylactate synthase. We did a lot of work. It's recently published. We show that this compound actually doesn't inhibit plant acetylactate synthase, but it's a reasonable inhibitor of fungal acetylactate synthase, especially the pathogenic fungi such as Fusarium species. We solved the crystal structure of this compound in the active site of a fungal acetylactate synthase, revealing a new mode of binding compared to other synthetic inhibitors of ALS. And what we also show is that in the when you deplete the fungus of these branching amino acids, this compound can actually be a reasonable inhibitor of Fusarium growth in liquid culture. So we think that Harzianic acid might play a role in the soil to inhibit fungal growth through the inhibition of the branching amino acid pathway. Okay, so that's all I want to talk about. This target of genome mining is being applied in different fashion, and that leads to a startup company in the Bay Area, Hexagon Bio, that's using this approach to find new medicines. Oops. Okay, so I think I will stop here, and thank you very much for your attention.
Great, thank you so much. Yeah, that was terrific. We've got a few questions. Let me get to those for you. The first is around heterologous expression of biosynthetic gene clusters. You shared a couple of examples, one in Aspergillus niger, the other in yeast. Could you share your perspective on how effective a tool that is for discovery?
Thank you for the question. So for us, this is extremely effective. In fact, it's our main weapon to reconstitute biosynthetic gene clusters. We still do a little bit genetics in the natural host, but these are often challenging, and many times the hosts are not available to us. Either they're derived from pathogenic plant that we need the permits for, or just not accessible. So we use this host, the Aspergillus nigelens host, extensively. There are other hosts like yeast, Saccharomyces cerevisiae, that we started in the beginning of our efforts, but it turns out that host is just not sophisticated enough, if you will, to handle a lot of the higher fungal enzymes. Yeah.
Right. Okay, second question. In the cannabinoid precursor clusters, is there a phylogenetic linkage between the HRPKS and the SAT domain of the NRPKS that explains the specificity / promiscuity of the alkyl moiety?
That's a great question. So we've looked into that a lot. Obviously, if we have a very high producing C8 producing pathway. It would be nice if we have an equally high producing C6 producing pathway to olivatoic acid. So we've looked into what determines the chain length specificity. As the question raised, the SAT domain at the NRPKS certainly governs chain length selection. And when we did the mix and match of different HRPKS with different NRPKS in Aspergillus nigelens, we find that the non-reducing PKS determines the chain length of the products. So that, I think, is determined by the SAT domain in selecting which C8 or C6 precursor from the HRPKS.
And we have a third question here. Fungi might be resistant to the compound because they don't have or express the target, for example, penicillin. Any idea how to screen or target for these activities?
That's great. That's a great question. So that's one thing we wonder too. I mean, we would love to be able to, even without these resistance enzymes, be able to predict what type of activity. Like I said, one of the example I showed, we were just frustrated. We don't know what the reason is, what the activity is for that very interesting looking compound. So I guess my answer would be that you can put it into a compound library, and hopefully one day somebody will pick it up in their screening, or you can go through the gambit of all the different assays that you can think of: antifungal, antibacterial, anti-cancer, and see if something hits. And that's a classic question, right? I mean, genome mining, the drawback of genome mining is that it's not, the cluster. Not the biological activity guided genome mining is that it's not activity guided. So right. What you find is what you find. Then you have to figure out what it does.
Yep. All right. Well, thank you